Okay, let's get back to the final session. So we have a broad session title of brain behavior. And again, I'm going to not give everybody long introductions because I'd rather let them have time. So please, Oksana, please take it away.
Sounds good? Okay. Good morning. I work in Palmer Lab, but I'm not going to talk about HS rats. I will talk about CFW mice instead today. So we looked at the hearing loss in this population. So, okay. This work has been done in the lab of Dr. Rick Friedman, who is a pioneer in the genetics of hearing loss. They did phenotyping, and we did and the Palmer Lab did sequencing and genetic analysis. Why care about hearing loss? Because it's common, it's prevalent, and it's especially bad in the older population. You can see that at the age of over 90, most people have hearing loss, and a lot of this hearing loss is actually age-related. It's not congenital. What's known about genetics of hearing loss in humans? So there were twin studies which found a very high heritability for hearing loss. There were a lot of genes which were identified for monogenic deafness. If the gene is not working, a person will be deaf. But most of it is not related to age-related hearing loss. The first human GWAS was done by Dr. Friedman in 2009, and there has been a few human GWAS performed since then. And the genes which are found, the QTLs which are found, have genes which overlap with mouse ontology related to inner ear pathology. So mouse models seem to reflect same biology as humans. So we can use mice to study. So what about mouse genetics? There were just a couple association studies in inbred panels, both performed by Friedman Lab. And there are also knockout mouse project, which identified a number of novel genes. But again, this is congenital, and this is in young mouse. It's not really related to age-related hearing loss. So surprisingly, this work is the first GWAS in outbred animal model. Study design. We used CFW mice because they are readily available. They are relatively cheap to buy and age, and they have good rapid decline in linkage disequilibrium. The target sample size is 2,000 animals. Right now, we're half through this number of animals, so we will. So I'm showing you the intermediate results. Genotyping. We use basically same genotyping that we use for HS rat. This is a very low coverage whole genome sequencing. But for HS rats, we have founders. And we use founders as reference. For CFW, we don't have founders. So the clever step here was to use a different population of CFW mice, which was also low coverage sequenced several years before, and published and on SRA. So we just took all this low coverage data and constructed a reference panel from over 3,000 mice, and it worked. We used Stitch and followed by Beagle for imputation, filtered SNPs for quality and being a common SNP, and ended up with more than 2 million autosomal SNPs with very low error rate, which we used in this study. The phenotype. So as I said, there's an overlap in biology and genetics in mice and humans. So we hope that there is enough variance in CFW mice to measure for age-related hearing loss to be variable. So you probably all experienced measuring hearing. It's when a nurse plays a tone and asks, can you hear it? And then if you can hear, you click, you say, yes, I can hear it, right? And if you don't hear, they increase the volume, and they increase the volume until you can hear. And this is your hearing threshold. What is the lowest volume sound in specific frequency that you still can hear? Yes, this is our phenotype, but there are some patients where you cannot ask, can you hear? Right, so instead of asking, we're measuring the response from brainstem, basically the neurons, the neurons responding to hearing, and this is what we're measuring. And this is a beautiful picture of hair cells, which are mechanoreceptors in our cochlea and in the mouse cochlea, which pick up the signal, right? This outer hair cell, inner hair cells, very beautiful. And this is our data set. You can see that we measured the hearing threshold on the y-axis across multiple frequencies on x-axis. And we measured it in very young mice, just a month and a half. This is a threshold in young mice in blue, in 6-month-old mice, and in 10-month-old mice. Right? And you can see the threshold lifted across all frequencies. This is a hearing loss. You can see the sample size. It's not too large at this point, but we are halfway through. And this picture demonstrates that in multiple frequencies, in different ages, sometimes mice are just deaf. They cannot hear. This number of 120 means that they cannot hear even at very loud volume. Like 100 decibels is like an airplane. It's really loud. So this is our second phenotype. Can they hear? Yes, no. All right, heritability. In human studies, heritability estimates were very wide, between 0 and 55%. There was one mouse study which estimated heritability, it's very high, 50-70% in BXD mice. What we can see is somewhere between 0 and 27, not very high. And this is GWAS. So we was relatively small sample size. We got several QTLs related to individual hearing loss at individual frequencies. And look at this beautiful QTL at chromosome 1, where mice are deaf at 10 months across all frequencies. Right? So I'm going to zoom in on this QTL. It's quite narrow, and it's over the gene for guanidine exchange factor 4. And this is this gene encodes a protein which participates in signal transduction and CDC42 pathway. This is a pathway which regulates cytoskeleton, and specifically actin cytoskeleton. Right. So if you look at this image of hair cells, hair cell which is labeled for actin, you can see that stereocilia, the mechanoreceptors, are basically made of actin. So it's not surprising that we are finding the gene which is involved in actin assembly. However, this gene has not been known to be involved with any type of hearing loss or vestibular, it's like another mechanoreceptors loss in mice or humans. So this gene is novel finding. So is it surprising? Is it is not surprising, but it's novel, right? Interestingly, there is another member of the same gene family, guanidine exchange factor 6, that is known to cause hair cell stereocilia loss. You can see like normal stereocilia here for wild type, and all rugged and broken stereocilia on the knockdown mouse on the left. And you can see that hearing threshold shifted in knockdown mouse, right? So it's, this other family member is known to be involved. This family member number 4 is a novel gene. So it looks like we discovered a novel gene with just small sample size. Another thing that I wanted to touch is the replication consideration. There were 2 studies in BXD mice which identified loci at chromosome 16 and 11. And the question, are we finding the same loci? Probably, right? They're not, they're reaching the significance. Maybe they will be significant later. And also we hope if they're significant, we'll be able to narrow down to like smaller, more narrow region. Future work, we will finish collecting the data. We'll perform genetic analysis and use a proper permutation test to determine the threshold, because the threshold that I showed was just arbitrary guess. We will be more creative about phenotypes, probably do PCA across all frequencies, and probably try to do structural equation modeling to look at the underlying genetics. And very exciting, the Friedman lab is collecting 1,000 cochlea inner ear samples, and they will do single cell analysis. So we'll do EQTL on that, EQTL analysis on that. With that, I will take questions.